So hi guys, my name is ONGRIA from STEM 1205, and today we are making animal cell.
So, here's the materials that we are using for today. First is, we're gonna use an 1/4 illustration board. Next, we're gonna use a scissor and a ruler. Next, we're gonna use a glue stick for our adhesive. And we're gonna use different colors of clay for our 3D diagram of animal cell. And lastly, we're gonna use the top of an old cardboard box. And for the making of the 3D animal cell. After outlining the body of the cell, on this part, I'm doing the nucleolus and the nucleus, and then the smooth ER and the rough ER. On this part, I'm already doing the GOLGI apparatus, which is the orange clay. After putting the GOLGI apparatus, I'm already doing the GOLGI vesicles. After doing the GOLGI apparatus and the GOLGI vesicles, I am molding here the mitochondrion and the microtubules. After putting the microtubules, I'm doing the cytoplasm, then the centrioles. And lastly, I already put the ribosomes. On this part, I'm putting the 3D cell diagram on the 1/4 illustration board. And after, I'm doing the different parts of the cell and its label. And now for the final product. The parts of animal cell. Cytoplasm, the semi-fluid substance of a cell that is external to the nuclear membrane and internal to the cellular membrane, sometimes described as the non-nuclear content of protoplasm. In eukaryotes, the cytoplasm contains all of the organelles. Rough endoplasmic reticulum, series of connected flattened sacs, part of continuous membrane organelle within the cytoplasm of eukaryotic cells that plays a central role in the synthesis of proteins. Smooth endoplasmic reticulum, meshwork of fine slide tubular membrane vesicles, part of a continuous membrane organelle within the cytoplasm of eukaryotic cells that is involved in the synthesis of the storage of lipids, including cholesterol, phospholipids, which are used in the production of a new cellular membrane. Ribosome, a sphere-shaped structure within the cytoplasm of the cell that is composed of RNA and protein and is the site of protein synthesis. Ribosomes are free in the cytoplasm and often attached to the membrane of the endoplasmic reticulum. Ribosomes exist in both eukaryotic and prokaryotic cells. Cell membrane is a thin, permeable membrane that surrounds the cytoplasm of the cell. Its function is to protect the integrity of the interior of the cell by allowing certain substances into the cell while keeping other substances out. Microtubules are longer filaments that are constantly assembling and disassembling. They play a crucial role in moving the daughter chromosomes to the newly forming daughter cells during mitosis, and bundles of microtubules from the cilia and flagella found in the protozoans and in the cells of some multicellular animals. Mitochondria or mitochondrion are membrane-bound cell organelles that generate most of the chemical energy needed to power cells' biochemical reactions. Chemical energy produced by the mitochondria is stored in a small molecule called adenine triphosphate or ATP. GOLGI apparatus or GOLGI body is a cell organelle that helps process and package proteins and lipid molecules, especially proteins destined to be exported from the cell. The nucleolus is a region found within the cell nucleus that is concerned with producing assembling the cell's ribosomes. Following assembly, ribosomes are transported to the cell cytoplasm where they serve as the sites for protein synthesis. The nucleus is a highly specialized organelle that serves as the information and administrative center of the cell. Most of the nuclear material consists of chromatin, the unstructured form the cell's DNA that will organize to form chromosomes during mitosis or cell division. Centrioles are paired barrel-shaped organelles located in the cytoplasm of animal cells near the nuclear envelope. Centrioles play a role in organizing microtubules that serve as the cell's skeletal system. They help determine the locations of the nucleus and other organelles within the cell. And lastly, the lysosomes are membrane-enclosed organelles that contain an array of enzymes capable of breaking down all types of biological polymers, proteins, nucleic acids, carbohydrates, and lipids.
And again, my name is ONGRIA from STEM 1205 with my animal cell 3D diagram. And that's all. Thank you.